So during the next few minutes, during the next few slides, I would like to introduce you a little bit to how we are doing research in a pharmaceutical company. As Andrew said, I'm working in Switzerland with Roche, but I think it's independent of the company where I'm coming from. I think it's quite similar, quite comparable how we work in the pharmaceutical industry. So the presentation is about how we do research and development. I think if you go today to a drugstore, you still see that there are numbers of drugs that are sold, but there are still many unmet medical needs. And along these lines, the pharmaceutical industry is still very busy in trying to find new treatments, new drugs, new medicines. And here you see two examples, like Xeloda, like Pegasus, two drugs that are on the market. And during my many years at Roche, I'm now almost about 20 years in this pharma R&D business, I've seen many fascinating projects, all aiming to have an improved health care, to find a better quality of life. That's a driver for us. That's why we go to work every day. How could this be looking like, this process of research and development? I think from the non-expert, the person that does not live in this pharmaceutical industry, it could look like that we have that there are somehow some bioactive compounds, like these powders that you see here on the screen, and then people usually think that we do afterwards some testing, expose them to some mice or so, and then afterwards, after having observed the benefits, for example curing diabetes in this mouse, we wrap this powder into a tablet, and then it looks like that, formulated, and afterwards we get the big money out of that. That's the way how one could perceive how we work. Unfortunately, it is a little bit more complicated. It doesn't work like that. In the end, you will see in this presentation that it's a highly complex process. It's a multidisciplinary process. So we have people from various disciplines in natural sciences, but also from other disciplines. It's a process that involves novel technologies that have received maybe a few years ago a Nobel Prize that were implemented. So it's a high-tech business, definitely. This is not for free to implement this high-tech. So it's combined with high costs. It's a long-term adventure. You will see during this presentation how long it really takes. So many years, I can tell you. And it's a high risk. We often are in a situation that we have to take decisions, that we are making sometimes the wrong decision. We hope that we most of the time do the right decision and bring the project successfully forward. Successfully forward, yes. Some of the projects make it to a drug on the market, but many of them fail, as somehow illustrated with this presentation, or with this slide here, where you see here a number of projects moving forward but face some stops, where there is a no-go and no way forward. How do we distinguish research and development? I think it's important to give you first a flavor of what activities are included in research and development. As you see here on the left-hand side, everything starts with an idea, and the aim is to have in the end a new innovative drug on the market to the benefit of the patient. The first part, what we call R, is research. The second is development. Research starts in essence with a concept. We test a target, we test an idea in a first, concept testing, and if there is a first impression that this might be a successful approach, for example, as I mentioned before, to treat diabetes, then we initiate the project. The project is then comprising a team, maybe initially 2-3 people. In the end there are maybe 50 or 100 persons involved that spend all their day on working on this project. The main task in research is to come up initially with a lead molecule, a molecular structure that leads us, therefore the name lead molecule, that leads us afterwards through the process to find optimized molecules. And after a number of years, I will come back to that later, we have identified something that we call a drug candidate, a molecule that looks good enough to become later a drug. That part here is still all preclinical. Everything in the microtiter plates, in the in vitro, how we call it, or in vivo, which means we test it in animals, but nothing in clinical trials. Then there is an intermediate phase where we prepare for clinical testing. That's where we do some toxicity testing to make sure that once we want to expose healthy volunteers to a drug, that they don't face a risk. Because I think that's the right of every healthy volunteer, that we make sure that this drug is safe. So here preparation for clinical tests, and then afterwards is quite a long phase for clinical development. Usually there is a distinction between phase 1, phase 2, phase 3. Phase 1 is where we test the compound in healthy volunteers. We try to find out what is the maximal tolerated dose, so we escalate the dose and see at which level do we see the first adverse events. Could be an itching, could be that some parameters in the blood is different than in the healthy population. And, oops, what happened now? It's coming back, good. So that's what we do here in phase 1, to test what the safety of the molecule is. In a phase 2, we then test the compound on a diseased person, so on a patient. So for example, to stick with diabetes, on a person that has diabetes. And there we want to see if the molecule is acting at all. Because what we have observed here in preclinical trials, in animal models, is this really translating into the human situation. Maybe we can cure a mice, but it's not effective in man. And we realize that only here. And in phase 3, we then go to large populations. There we talk about clinical trials that comprise more than 1,000 patients usually. I could mention that currently in Roche we have a clinical trial ongoing that comprises 15,000 patients. 15,000 patients that receive a drug during 3-4 years. So this means that if you look at this graph here, an increasing number in the costs. So the costs are here relatively low, but here it really is skyrocketing. And not only for us, but I think for the whole industry, we're talking now about 1.3 billion US dollars that are necessary to bring one drug to the market, to launch it. We don't know if it's afterwards a success. We don't know if it's a success like an iPhone or iPad. We just don't know. But we first have to invest this amount of money. And I think we can only do that if our drugs on the market generate some revenues. Without that, there is no sustainability in this R&D process. Now, this is I mentioned to you before research and clinical development. Here is another way of depicting it. You see here again phase 1, phase 2, phase 3, and so on, and here the preclinical phases. If you want to bring one drug to the market, you can imagine, or I mentioned to you before, that in the previous phases there is attrition, so some of the molecules will not make it to the market, and the numbers are somehow as follows. So we need to enter with more or less 10 molecules into clinical trials to be successful with one that is being launched on the market. So only 1 out of these 10s are making it. And now you can imagine that here in the preclinical part we even have to start with more molecules, with more projects. If you look at these numbers here, I've changed here the color of the bars here from black to white because these are drugs or molecules and these are projects. So we need to start with something like 60 projects that lead then in the end to one drug. And these 60 project teams, they synthesize something like 10,000 of molecules. So only something like 10,000 out of 1 is making it to the market. Of course this depends, this is an average. In some indications it's even worse, like neurosciences, Alzheimer's diseases, Parkinson's and so on. In others it's the numbers are a little bit better. But nevertheless the picture, this is here the average, is somehow like that. And to start a project, we before have probably usually something like 10 ideas. So scientists came up with great ideas what one could do, and only then afterwards 1 out of these 10 is then selected for a project. The red bars indicate milestones, so we have checks where we look at if we have achieved a certain quality in our project and only then move forward. I mentioned to you before the number of people. Here's another figure, I think it's the same ballpark figure. 100 to 300 people are needed to generate here 1 of those clinical candidates per year and to bring that forward. And out of these 1, 0.07, it's the same number as you see here below. So quite a high risk, as I mentioned to you initially. If you look at the time frame, then we talk here about something which is more than 10 years. It could be 12 years, it could be 15 years. I know very precisely because I've been involved in such a project. We started in the year 2010 with a diabetes project, which is now in phase 3. So not yet on the market, 12 years ago. Represents almost exactly these numbers here. So you see the numbers, the years that it takes in clinical and in preclinical. And before that, before we start here with the project, there is where this ID generation is taking place. This takes another, I wrote here 2 years, could also be longer. Maybe in our organization, maybe in academia, somebody has found a great way to move forward, and to bring it afterwards to the market, it takes more than a decade. That's more or less it. And since we are talking here about patents, usually we file patents in this phase here, in the lead identification phase. There is a balance. On one end, we would like to patent as soon as possible to make sure that we have the freedom to operate, that not suddenly a competitor is filing a patent for the same drug, for the same substance class. So we want to patent as early as possible. Just 1 second and I'll come back to you. And on the other hand, we want to delay patenting as long as possible, because of course we want to have, once on the market, a coverage which is as long as possible. You had a question?
Yeah, a very small clarification. If you could explain that again to me, 100 to 300 people needed in research to produce 1 clinical candidate per year?
Yes. So if we have here, if we want to achieve 1 clinical candidate in an organization, I'm working in Roche at Basel in Switzerland. And in this organization is asked to deliver 1 clinical candidate. So 1 out of these 15 molecules here that then make it to 1. If we are asked to deliver 1 of these clinical candidates, then we need to have in the background something like 100 to 300 people. That are people in chemistry, in toxicology and so on. I'll come to the disciplines, what they are doing very precisely in the next slides. Okay, so 10 years patent life, as you have usually already expired when we are entering the market. Just a second ago I mentioned to you the disciplines that these 100 and 300 people are working on. Simplified view is chemistry, those that prepare these powders, the biology that is doing some tests in the tube, in the microtiter plate, so in vitro, and then the person that does pharmacology, so studies in animals. Nowadays it has changed. We don't have only these disciplines, we have split it up. There are more newers have been showing up, like genomics a few years ago, where people started to sequence the DNA. We look at biostructure. We look at we need informatics to connect all these disciplines, to generate, to integrate all the data. We look at metabolism of the drug. So if the drug is taken up by the body, how fast it is, how is it secreted, and so on. So it has become a highly complex network of various disciplines. And the 100 to 300 people I mentioned to you before are working in these areas. And I think that's the fascinating thing. I still like to work in this environment because it's so interdisciplinary, but it's also very complicated to bring something forward. And you can imagine, since these guys are not just using a pencil and a sheet of paper, but some equipment, like they go for gene expression profile, they have here a biotechnology process to ferment, they have here maybe an ultra high throughput screen that allows them to test 1 million of compounds in 2 weeks on a certain assay, for example, or they do X-ray structure where they need, of course, also an X-ray beam, like here in Geneva at the CERN, for example. So it's a highly expensive adventure that we are undertaking here. I don't want to go into details of this technology unless there would be some questions afterwards. And there is another level of complexity. Here you see another way of presenting here the different phases, phase 1, phase 0, phase 1, and so on, phase 2, phase 3. One phase in between is called lead optimization. I mentioned to you we have at one point a molecule that we want to improve, to optimize. For example, it is perfectly working in an animal model, but only if you inject it constantly as an IV infusion. But in the end, the patient wants to swallow a tablet once a day. So we need to improve the bioavailability, that it's really taken up. If I swallow the pill, that it's afterwards through the stomach taken up into the body, it's afterwards rightly distributed and in the end secreted. So we need to optimize the molecule. Or we want to have the molecule optimized towards selectivity so that we reduce the toxic effect of the compound. So usually the way it goes is that the chemist has synthesized the molecule, we test it in an assay, we develop what we call a structure-activity relationship, and then we design new molecules. And in this circle, the chemist or the project team optimizes their molecules. Now before I mentioned to you a couple of ways to optimize bioavailability, tissue distribution, selectivity, so there is not just one assay, but there are multiple assays. Means we have a multi-dimensional optimization. Makes it more complex, takes longer, but in the end we hope that it's better to do it in parallel than sequentially. Now, why so complex? Has it changed? Has it changed in the recent years? I've here 3 points that I would like to mention. One is low-hanging fruits are gone. I think the diseases like finding antibiotics, where you in essence want to kill a bug that is not a human being, that's relatively easy. It's easy to kill something. But it's much more difficult if you want not to kill a bug, an antibiotic, but maybe a cancer cell. The cancer cell is very close to the human situation, to the human healthy cells. So there, it's much more difficult. Or if you talk about metabolic diseases, like diabetes, You just want to have a balance that is no more appropriately balanced, and you want to return it back to normal. So it's not about destroying, but bringing it back to normal. That's inherently more complicated, as everybody knows that wants to fix his car, for example. And along these lines, we're working more on these more difficult tasks, because the low-hanging fruits, we have already generated solutions. So this poor scientist is now in the difficult situation that he has no more low-hanging fruits. Now this helicopter here is maybe representing the technology that I've shown you before. It's another way to approach the more difficult targets. There are also increasing demands. Type 2 diabetes. In the past it was okay to show that the glucose levels in the blood are lowered. And after a certain period of time, the healthcare authorities said, who cares about blood glucose in your blood? In the end, I want to live longer, and I don't care about the value of the blood, of the glucose in the blood. So we need now, since about 5 years, we need to show that the patients really benefit on mortality and morbidity. And this is the consequence why we then have such long studies or expensive studies that I explained to you before, like 16,000 patients treated during a number of years to show not only that cholesterol is lowered, but that the people live longer. So the increasing demands, I think it's right from the healthcare authorities, from the society, but has an impact. Natural products, a topic that you're discussing here in this symposium. They are scientifically of value to explore a new target, to work with them. You're all well aware of that. But I have here a structure depicted of one of them, of one of such a natural product. And for those that remember a little bit their chemistry courses in high school or during study, it's a complex structure. And to synthesize such a structure is very complex. To do that. So the optimization cycle I showed you before is then getting longer and longer, more difficult and more difficult. And this is telling us that we often don't want to do that because it's so inherently difficult to optimize. For example, to optimize the oral bioavailability or the PK, which was exactly the case for this molecule. It worked nice in the Petri dish, but no more in the animal. The complex synthesis could also mean that it's afterwards expensive to synthesize. And another point is, of course, the reliability of the supply. If this compound is only found in small quantities on our planet. So with that, I'm on the last slide. I think there are still many unmet medical needs, as I've mentioned to you. It's complex, it's frontier science, high-tech, expensive and high risk. And here you have some numbers that it takes, for example, more than 10 years from project start to entering into the market. Only 10 years, less than 10 years remain probably as patent protection once we are on the market. And the costs to bring one molecule to the market is in the range of 1.3 billion US dollars. And with that, I would like to thank you. I don't know, I think questions are coming later.